Okay, so in this video, we're going to be going over the phases of meiosis and really what you need to at least be able to understand in terms of the point and really the ins and outs of what's going on. So, um, the first thing that you need to know about meiosis, like I mentioned, let's kind of get the generics out of the way of really what you should be able to understand. Now, understanding a few things here is that the point of meiosis, okay, the point of meiosis is to do sexual reproduction. So the point of all of this is sexual repro. Now, other things, if you're doing meiosis, kind of bigger picture things, is you're going to make 4 gametes, and they're going to be non-identical. Okay. Also, in terms of that, the gametes that get produced are going to have 1/2 the genetic info as the parental cell. Okay. So we'll kind of go through the ins and outs of each one of these, but let's at least kind of take a look here of what you should be able to understand is this is typically what a cell is going to look like. The only cells that do this are going to be your sex cells. So things that are going to be actually producing your gametes. So testes in males, the ovaries in females. But again, plants also do sexual reproduction. A lot of species are able to do sexual reproduction. So this is kind of a generic thing. You're always going to produce 4 cells. Okay. Now, again, these cells at the end, depending on the species type, they have to actually do some type of fertilization. So what comes after meiosis? It's typically going to be fertilization. Okay. Now let's look at this first. Interphase. I highly advise that you go back and recheck the cell cycle question because, or I should say, the cell cycle video, because how is this controlled? It's going to be the same. So a couple of things that I need to emphasize here. This all kind of all scribbled up. That's typically how your DNA looks the majority of the time. Only when it's going to be doing some form of cell division do you get the looks of chromosomes looking here. Okay. Now, in interphase, let's kind of talk about the generics, right? Your DNA has to replicate. Don't forget the cell cycle. So it still has the G1, the S, then the G2. So that happens in both meiosis and mitosis. So most of the time it's just taught with mitosis, but certain indicators are going to work the same for meiosis as well. Okay. So before any type of cell division, you always have to replicate your DNA. So I really need to make sure you guys feel okay on that. Right? Now, meiosis is really going to be broken up into 2 main steps. It goes meiosis 1 and meiosis 2. Like I mentioned in the last video, yes, it's prophase, metaphase, anaphase, telophase. That happens twice. But there's some pretty significant differences that you should be able to understand and really just understand the bigger picture that the point of meiosis is far different than the point of mitosis. Mitosis is just replicating identical cells, growth and repair. Meiosis, you're doing meiosis strictly for the point of sexual reproduction. So I'm going to start off by going through some notes that I had made earlier and you guys had seen previously while I was gone. I'm actually going to keep that. So let's jump here. Now, we got to start off talking about a couple of things here, and this will help for the next chapters as well. Now, we're going to talk about what are homologous chromosomes. I mentioned it in the last video, but this one is a little more generic. So I shouldn't say generic, a little easier to see. So homologous chromosomes, those are going to be defined by chromosomes that have the same genes. So again, what is a gene? It's a region of DNA that codes for a protein. So if I looked at these 2 chromosomes, I would say that these 2 are homologous. Okay. So when we refer to them as being homologs, okay, it has the A gene. This one's dominant, this one's recessive. These 2, these would be homozygous for big B, big B. These would be homozygous for little b, little b. Okay, so, and we get into that word now, allele. That's a form, dominant recessive version of a gene. So I would have a dominant version of this gene here. I would have a recessive version of this gene here. So I just want you guys to understand that. So these 2, they're the same type of chromosome. They're different. Maybe this one I got from the mom. Maybe this one I got from the dad. Okay. but regardless, they're homologs, okay? So that's what we're getting into in terms of how these things get passed down, all right? So, and this is typically what a cell looks like. So let's kind of get going here. So before we get into it, let's remind you guys of ploidy. N is the number of types of chromosomes. So it's really this simple equation. So most cells are diploid, di meaning two, in somatic cells, which again, somatic cells are your non-sex cells, okay? They're gonna be haploid in sex cells after meiosis. So for example, if this was my my starting parental cell, right? If this would be my parental cell right here, 2n equals 8, that means I have 2 of each type of chromosome, and in this case would equal 4. So if it asked you, what is the ploidy state after meiosis? Well, it would just be 1n equals 4 after meiosis, and that's what we're going to show you in this video. So just understand that. But full disclosure, you got to understand biology is not limited just to humans. There's all kinds of weird genetics, especially when you get into plants. Things like, well, let's look at rice first. Okay, rice would be 2n equals 24. They're using x as their variable. So they have 2 of each type, it's diploid. Now, soybeans would be 2n equals 40, so they have 2 of each type. But then you can get into some weird fruits like things like bananas. They're actually going to be triploid, so they would actually be 3n equals 33. So if it asks you, like, how many types of chromosomes do bananas have? They have 11 types, but they have 3 of each type in each cell. Look at sugarcane. That's crazy. That's been selected for quite a while, where it's actually 8n equals 80. So 80 chromosomes in the nucleus of one of those sugarcane plants, but they have 8 of each type. So don't think of everything as just being diploid. They could throw out some weird questions on the genetics of different plants. So, but most of the time, I would predict that they'll give you something that's diploid. But just don't limit it just in terms of your understanding that every cell is diploid or haploid, because they're not. So kind of getting this point across again, what is the point of meiosis? It's sexual reproduction. If you're doing meiosis, even if you get one of these weird, funky ploidy states, they're always going to contain half of the genetic material, okay? And this allows the cells produced to fertilize with another to make a new organism, okay? So let's get into this, and we'll kind of show you some visuals jumping back and forth, okay? Now, I like to kind of start this off by showing you a couple of things here. Well, this is the cell. I'm saying that here's the nucleus. Now we could have all kinds of other stuff here, like, you know, the Golgi apparatus, ribosome, whatever have you, but we're just going to be focused on the nucleus, and then this would be like the cell membrane. So don't fixate if it's a plant or animal cell. We'll just say right now that this is just a simple version of meiosis, okay? Now let's make sure you understand what this is talking about. 2n equals 4, where that n stands for the number of types of chromosomes. 4 is the total number of chromosomes. And notice that this is showing you that the DNA has not replicated yet. So let's get into this. So I have my alleles. I hope that you guys understand that these two are homologs. Okay, it has the A gene, this has the B gene, this has the C gene. So I would be heterozygous for A, heterozygous for B, and it looks like homozygous recessive for C. Okay, these two, hopefully you can understand, are also homologous. Okay, I have the F gene, I got the G gene. Don't worry about what they code for. I don't know. This is just showing you a random representation of it. F and F are going to code for a protein. I've got a dominant version and a recessive version. So if I asked you how many chromosome types do you have in this cell, hopefully you would be able to tell me you have 2 types of chromosomes, but you have 2 of each type. So that's where this ploidy state is coming from, 2n equals 4. Okay? Now, as I mentioned before, any type of cell division involves one key component, and that is going to be your DNA has to replicate. Okay? So basically, if I were to go through this and make sure that you guys can do this as well, I'll try and keep the colors consistent. So if I were to replicate this, your chromosome now, if I made an exact copy of this chromosome, okay, I want you to understand that it would look like this. This will be A, this would be B, and this would be C, okay? I would do the same for all of these, right? And hopefully now that it's towards the end of the year, we're just kind of reviewing, remember, it's DNA replication, right? Helicase opens it up. You have your topoisomerase preventing it from opening the straining, all those enzymes that you studied, right? So DNA polymerase 3, you got your leading in line, all that applies right here when you replicate it. So just remember the big picture stuff. So basically this is showing you after the DNA has replicated. I've copied these things. They're perfect in terms of they've replicated, right? So in interphase, that's just showing you, remember, the S phase is where all this takes place, okay? So the S phase of the cell cycle goes G1, S, G2. S is standing for your synthesis. That's where all of this takes place, okay? Now, we're still keeping the ploidy state 2n equals 4. That's going to stress some of you guys out, but just understand you've just made an exact copy. You're not changing the ploidy state, and a cell doesn't exist like this for very long. Once your DNA replicates, it's almost immediate that it's going to start the division shortly after. So don't let that stress you out. So let me, I'm going to keep this real quick. Let's show you a visual here. There's a few things that my video doesn't show. So let's kind of take a look here. These are talking about your nuclear envelope is going to get broken down. These are showing you your centrosomes. They're basically extending out your microtubules. They're going to attach to the chromosomes themselves. So let's kind of do this. We'll take it slow. Like mitosis, meiosis is preceded by interphase, which includes the chromosomes replicate. Okay, so your centrosomes are basically duplicating. These are your microtubules. These are going to provide the tracks for these chromosomes to ride on. That's important for you guys to understand here. Okay, so in terms of that, yeah, I don't want to show you prophase quite yet, but let's go to this one in interphase. I'm just going to jump ahead real quick because they show you some interesting facts here. I like this one a lot. So basically they have it color coordinated to where, let's say the blue one is the mom, the red is the dad. It's showing you DNA replication here. These two, they're trying to show our homologs. And it's basically kind of breaking a few things down here where, remember that DNA, if it's eukaryotic and they're wrapped around those nice little histone proteins, I would call this chromatin. As your DNA condenses down, we're going to call these chromosomes. So kind of different versions of what's going on here. So now when you look at it from this standpoint, so like I said, we've replicated your DNA here. Now we're actually going to get into really meiosis 1. Okay, so we think about what are the phases of meiosis. It's going to be prophase, metaphase, anaphase, telophase. That's going to be meiosis 1. And then you got prophase 2, metaphase 2, anaphase 2, telophase 2. I'm going to be honest, your AP test typically isn't going to test you any specifics on each individual phase, but at least follow through with the gametes and the alleles and what's going on here, and you should be in good shape. So let's talk about what happens in prophase 1. I'm only going to hit the key points of what you should know is if it happens in your AP test. So prophase 1 is a big one that you guys should mention. And the big thing about prophase 1 is that you want to understand crossing over occurs in prophase 1. Okay, now it's a little ahead of it, but I'm going to just tell you now. Prophase 1 is where you're going to basically alleles can be exchanged if they are the same gene. So that means I can exchange a G for a G. I can't exchange a G for an F. Okay, that would be a problem. Now notice you're also only exchanging the alleles at the tips of the chromosomes. The genes on the exterior aren't necessarily going to be participating in any crossing over. Okay. Now a lot of this is going to be completely random. It does happen quite a bit. So I just chose to cross over the G's. I could have, and you can practice this on your own. I could cross over these two together. I could swap over the B with that one. I can swap over the A with that one. But prophase 1, a big thing is that you're actually going to be creating recombinant I shouldn't say recombinant genes, but you're going to be creating recombinant chromosomes. So I'll just abbreviate it chromos. Okay. So prophase 1 is where you're actually going to be creating recombinant chromosomes. Now, if you don't remember what recombinant means, that means you're getting new combinations of genes on a chromosome. So they're different than the parental type. So recombinant chromosomes happen in prophase 1, early prophase 1. Okay, so this is just further common test question is when does crossing over occur? It's during early prophase 1. This occurs at the synapse. Crossing over can only occur between homologous chromosomes. So we're still in prophase 1. So I'm going to go and keep that. Let's show you the visuals here, what's going on. So in prophase 1, your DNA starts to condense, and that's when you actually start to see them as chromosomes. So that's where they look like that. And these are going to be your chromatids, okay? Now, if I didn't make mention of that, I will talk about it now. So if I refer to this, yeah, these two, once they're copied, are going to refer to these as sister chromatids. They're identical. They're identical because they're of the same chromosome. I wouldn't call this chromosome identical to this one. They're homologs, but they're not identical. When they've replicated, that's when we refer to these as sister chromatids. Sister chromatids. These two are sister chromatids. These two are sister chromatids. These two are sister chromatids. So I just needed to make sure I talked about that one. So prophase 1, let's get back to it. It's the condensing of the chromosomes. Homologous chromosomes come together in pairs. That's called your synapse, okay? When these two line up, I'm going to call this a tetrad. I would call these four a tetrad as well, okay? Chromatids of homologous chromosomes cross over each other and exchange part at chiasmata, okay? So in terms of that, let's kind of see if this one's going to show it. I haven't pre-watched this, but yeah, there's the chiasmata. Singular is going to be chiasma. Now, I don't think they're going to go showing, yeah, so they're showing just the centrioles extending out. It's going to help break down the nuclear envelope. So now we no longer have what's known as like, I don't want to say a nucleus, but your nuclear envelope is now broken. That's going to allow these microtubules to attach the kinetochore, okay? They're going to capture the kinetochore, which are basically these proteins on the chromosomes, and they can actually start, literally what they do is they're going to be pushing those chromosomes towards the metaphase plate, so towards this imaginary plane at the center. So understand that. Common things about prophase 1, we'll show you this one as well. So for prophase 1, there's going to be some interesting facts here. Here's your centrioles. There's your nucleus. Another way of seeing this. These are your microtubules. They're going to help break down the nucleus. So if we look at what's going on here inside the nucleus, here is where your DNA is condensed. We're going to call them chromosomes. That would be essentially your centromere. And this is going to show you your crossing over in prophase 1. So you've actually exchanged and crisscrossed, and DNA has jumped from one sister chromatid to the next if it's a homologous pair. So crossing over is key in terms of increasing genetic variance. So this is showing you those microtubules attach at the centromere, where there's one of those kinetochore proteins right here. And what it does is it's going to help push and force those chromosomes all towards the center. So that's something that is important. So let's jump back to this, and hopefully this will make some sense to all of us. So again, I can't physically exchange this genetic information. Crossing over takes place in prophase 1. Now, when these things all line up, that's when we're going to call them a tetrad. So your homologs line up in my metaphase 1 of meiosis 1. Okay. Now here's again your microtubules attaching here. It forces them towards the center, and that's what I'm telling you. Your homologs line up. Okay. Now there is one thing I got to mention here that is also going to be important for you guys to at least understand is the only thing that you need to understand about my metaphase 1 in meiosis 1 is homologs line up. Okay, that means the F and the G had to line up next to the F and the G chromosomes. The A, B, C had to line up to this A, B, C. Now, how they line up is random in the sense that I could have had the red on this side, but I could easily have exchanged these two. These two could have been flopped. Same thing as these two. I could have had 2 blue and 2 red. Okay, so that's what I'm showing you with these two pictures, right? It could line up like this, or they could line up like this. Both are okay. The only thing that wouldn't be okay is that if I had this chromosome lined up with the F and the G, that would not be okay. So just know that when we say homologs line up, that's what we're talking about. Now, again, I'm showing crossing over with the G here, but that's random. I could have chosen the C's. I could have flopped those ones over, but I have created a recombinant chromosome here. So let me just remind you of what recombinant means. Recombinant means that I now have a new genetically different chromosome, okay? So this is my recombinant. I have the little f, the big G. That wasn't like what the parent was. So that would be my new recombinant chromosome. So let's look at this one. These ones should all go pretty quick now. So in metaphase 1, of meiosis 1, you basically are going to force them all towards the center. Okay, so there's your metaphase plate connected at the kinetochore. So that's something that is going to be super helpful, is knowing that your homologs line up. So jumping to the other biofix, let's look at metaphase 1. And I think these ones go really, really quick. So yeah, all it's showing you is that the microtubules are forcing all of those homologs together. Okay. Now, that's important to understand, and I hope this is helping you kind of see the bigger picture. But what's going to happen next is, after metaphase 1, okay, is going to be anaphase 1. Now, there are a few things about anaphase 1 that you need to at least kind of understand. Is your homologs started to separate, okay? Your homologs are being pulled apart, right? And that is something that the microtubules are doing, okay? So it is still essentially going to be diploid. Your chromatids are going to be staying attached. So meaning I still have my sister chromatids attached, okay? I still have my sister chromatids here. I still have my sister chromatids here. So that's something that you at least want to be understanding, okay? So let's take a quick look here at the other visuals, and I will show you this clip as well, or at least this slide. So when you think about anaphase 1, right, like we just talked about, in metaphase 1, right, your homologs have lined up. I would call this a tetrad, okay? Tetrad, a tetrad, that's when your homologs line up, okay? When you're pulling them apart in anaphase 1, okay, your homolog chromosomes have separated. Your sister chromatids remained attached. Now notice that these two right here are showing some crossing over took place here. Some genetic information is crossed over here, okay? So in anaphase, you're pulling apart your homologs in anaphase 1. So we'll just show you here. This is showing you now anaphase 1. And in anaphase 1, big things here is that your homologs have separated. Notice I have a recombinant chromosome here, recombinant on that side, and they are now being pulled apart, okay? Now, to wrap this one up on meiosis 1, let's talk about what's going on next. Really common things is that in telophase 1 and cytokinesis, you now have 2 haploid cells starting to form. So why do I call them haploid? Well, notice that this homologous pair has been separated from that homologous pair. This homologous pair has been separated from that homologous pair. These are just identical sister chromatids. We don't call these homologs. These are sister chromatids. The homologous pair has been separated, so they're always haploid after meiosis 1. So this is showing you the 2 cells after meiosis 1. So your homologous pairs have been separated. Each cells are haploid because the homologous pair is now over here. Homologous pair is now over here. Now again, I have created one recombinant type of chromosome because I crossed these things over in meiosis 1. So they're now 1n equals 2, okay? So that is meiosis 1. We still got a little bit more to go, but the rest go really fast. So let's just show you this. After meiosis 1, let's show you this real quick. Note that each chromosome still consists of the sister chromatids, and your nucleus essentially reforms, okay? But I wouldn't necessarily call these viable cells because, again, these are just sister chromatids. This homologous pair is separated from that homologous pair. This homologous pair is separated from that one. So that's going to be happening during meiosis 1. We have now made 2 cells. But meiosis is definitely not over. So let's jump back to where we're at on this first screen here. Basically, what I walked you through is everything up to here. Okay, we did cytokinesis. Now we're going to do meiosis 1, where we're going to be pulling apart our sister chromatids. Now, this one goes quite a bit faster in terms of the explanation side of things because we don't need to get crazy specific. But basically, it's going to be prophase, metaphase, anaphase, telophase again. So it's going to be prophase 2, anaphase 2, or excuse me, prophase 2, metaphase 2, anaphase 2, and telophase 2. So basically, this is showing you in prophase 2, I don't have a slide here, but basically your chromosomes condense down again. They line up at the metaphase plate, metaphase 2. In anaphase 2, your chromatids start to separate out. So you're actually pulling apart your sister chromatids. So cell 1, cell 2, they're being pulled apart. So I'm actually separating out my sister chromatids, okay? Now, that's important to understand because then when you get to telophase and cytokinesis, here's what you really need to look at. if I pulled these two apart, these are going to be these two gametes that were produced from that cell. These two gametes are produced on that one. So I would want you guys to at least be able to understand and identify, okay, I have two cells that are not genetically different than the parent. So I have a recombinant chromosome here, and I have a recombinant chromosome here. So a couple of things that you really want to make sure you feel comfortable with is each cell contains only one type of each chromosome. So that means that I have the FG chromosome here, I got the ABC chromosome here, I got the FG chromosome here, I got the ABC one here. So it would be wrong if I had FG and then another FG. It would be wrong if I had an ABC and another ABC chromosome after meiosis II. So that's something that is a really common mistake, and that means you separated your chromosomes out incorrectly. So like I talked about earlier, recombinants are going to be cells produced that have genetically different chromosomes than the parental one. So these, like I said, that would be a recombinant one. This would be a recombinant one, okay? And this has occurred, I should say, this is because of what happened in prophase 1, okay? That is critical. It's because of prophase 1, that's what happened. So if you keep that in mind, let's kind of jump ahead. Let me show you this. Going to this visual, let's just kind of play the rest out. This should go quick. So we'll kind of go through all of this together now. In meiosis II, it's essentially the same as mitosis. The important thing to remember is that meiosis II begins with a haploid cell. So these things are already haploid. So notice now how they line up. They just line up at the metaphase plate, but these are your sister chromatids. So those are your two. Moving forward, during metaphase 2, the chromosomes line up towards the metaphase plate. In anaphase 2, the sister chromatids are pulled apart. And in telophase 2, notice what's going on here. There's a couple of things. In cytokinesis, your cytoplasm splits, your nucleus reforms, and there's going to be your 4 cells, okay, with half of them. So in terms of this, let's kind of take a quick second versus listing the difference. I want to see if there's another visual here. I don't think there is. So to kind of wrap this one up, let's show you the same biofix version, which is going to be, let's just look at meiosis II. So in meiosis II, again, you've already separated your homologs, so it's showing you the other cell over here. So those are all your sister chromatids. That's anaphase 2. You're pulling apart your sister chromatids. Then again, in cytokinesis, I should say cytokinesis 2, you get your 4 cells produced here at the end, okay, that are genetically different. So there's a lot going on to digest in meiosis II, but a couple of key reminders here is what happens if 2 gametes combine, right? So what I'm trying to say is, well, what is the point of meiosis? It's always to do fertilization. So it's random, but let's say if I had just this gamete right here, so I'll put a little star here. If these 2 recombined, notice now, if this gamete fused with this one, this is now my new cell that would be 2n equals 4. And this would essentially be the new offspring. So that's going to be the key thing here. This was 1n equals 2. This cell was also 1n equals 2. When these 2 combined, it became 2n equals 4. And that's the whole point, is getting those gametes fusing together. Okay. So summary, some key differences that you want to know. Synapse and crossing over takes place in prophase 1. We call them tetrads when they line up in metaphase 1. So that's when your homologs pair up. And your separation of your homologs is going to be in anaphase 1. So the last little bit is what are the really big things of genetic variance in meiosis? So independent assortment, basically, if I didn't mention that earlier, it's basically how those things line up in metaphase 1. So let me show you this. Right back here at the beginning, that's what I'm talking about. They could have lined up like this, so I could have the red and the blue here, or they could have lined up like this, okay? So that's that law of independent assortment, how those things line up, okay? Now, like I'm saying here, possibility 1, possibility 2, how they line up. Notice that even without showing crossing over, I get these 4 combination of gametes here, I get these 4 combination of gametes here based off of how these things lined up, okay? So Now, if I threw in crossing over, and this shows you that in humans it's 2 to the nth power. We have 23 types, so even without crossing over, you have 8 million combinations just by how they line up. You throw in crossing over, it gets ridiculous how much variance we could get. Again, how else do we get variation in meiosis? Crossing over. And again, like we talked about, that's going to be prophase 1. But the last one is random fertilization. That's kind of what I talked about at the end. This gamete could fuse with this gamete. If they came together, you would get a new combination of a cell, right? So I would get 2 that are going to be homologs for the longer red chromosome. I would get a short red and 1 blue one. You get the idea. So I don't know why this isn't going, but again, like I was mentioning earlier, is the point of that was just to increase the stronger traits are going to be the ones that are going to be surviving. So yeah, here we go. So like I said, random fertilization, it's all about the gametes that get produced and the gametes fusing together. If I took this gamete with this gamete and these 2 combined, that's your new parental cell, okay? So kind of last little bit of meiosis facts. Just to understand the basics. What is the point of meiosis? It's to do sexual reproduction. You increase genetic variance. And we'll see you guys in the next video.